Okay, a very good evening to everyone. So today we are going to start with our last chapter of the revision, that is chapter number 10, microbes in human welfare. Okay, so before starting the chapter, please let me know through this technical poll whether I am audible and visible to you or not. Okay, please participate in the poll so that I got to know. Okay. Okay, great then. Okay, so as the topic itself says, it is saying microbes in human welfare. So we are talking about the microbes. What is microbe? Can you tell me? What is microbe? What are microbes actually? Anyone in chat box? Minute organisms around us, small organism we cannot see by naked eyes. Very good, everyone. Very good. You have given a correct answer. Microbes means those organisms, or we can call them microbes, a micro organism, right? So microorganism means those organisms which are unable to be seen by the help of naked eyes. As you can see here, these are the microbes, but these microbes can only be seen by the help of the microscope. They are so smaller in size that we are unable to look at them in by the naked eyes, right? And we can say these microbes are present everywhere now in our surrounding in which we are sitting, in which we are present. That is itself contains millions, billions, and trillions of the microorganisms. But we are going to talk about the microbes or microorganisms which are helpful for us. See, when we put them on a slide and dish, right? Like for example, you have taken the curd. Okay, you have taken a small amount of curd. You know now curd is made up of which bacteria? Tell me, Nandish. Curd is made up of which bacteria? Labs. That is lactic acid bacillus, right? Lactic acid bacillus. So those are present throughout the curd. Okay, when you make a smear, that means when you make a thin slide of those curd, then you have to put some dye. Okay, when you keep it under the microscope itself has so many lens, and the lens have a power to magnify the image, right? You are using in physics, you have commonly use the magnifying glass, right, which you can use to uh, focus the sunlight and anything else, through which you can, if you have observed your hand, you will find that it is of very big size, right? So the microscope we have the lenses, and those lenses help in the magnification of the size, right? This is how just like the magnification or zooming in the picture. When we take the picture, we usually zoom in, so our lens focuses on that particular object. In a very same way, the lens which are present in the microscope zoom in, or we can say they magnifies the size of the object so that we can easily visualize them. Okay, is it clear, Nandish, now? Okay, so before starting the question, okay, let's discuss this. Before starting the session, let's discuss this question first. Which of the following is not related to the microbes? Okay, you have already studied about microbes a lot in your biological classification, chapter 11. And before that, you have done about the microbes in the class 8, right? So according to you, what is the correct answer? Your question is, which of the following is not related to microbes? Your option A says microbes are not ideal for biotechnology. Okay, very good. Everyone has already given their answer in the chat box. So, right, your answer is correct. You say they, let's see, your answer is correct or not. Okay, so option A says the microbes are not ideal for the biotechnology. They are not useful. If something is not ideal, it will not be useful. So we cannot say, right? We cannot say it is not ideal for biotechnology. They are ideal. Okay. So let's see another option. Maybe we found something good enough. Second option says the microbes produces chemicals during their metabolism, right? They produces chemicals, whatever enzyme they use to metabolize the substrate, the reactant is always in the form of enzyme, right? They can be a chemical. So this is correct. Option C says microbes can grow on artificial medium. Yes, they can grow on artificial medium. In labs, we only provide them the artificial medium, the controlled and nutritive medium, so that they can grow. And the option D says microbes can live in highly acidic medium. See beta, the microbes can also grow in acidic medium. Yes, you have also given your answer itself that the archaebacteria, and we have so many thermoacidophilus bacteria which can survive in acidic medium, right? And that acidic medium is very highly acidic medium. If we go for the initial primitive earth state, right, when no one was there, when no one was there, initially what happened? The microorganism, that is bacteria, formed. At that time, the oxides of nitrogen and sulfur were so high in amount that the water which is present in the pits, or water that get accumulated in the pits, were actually very acidic in nature. Okay, so we can say the microbes can live in highly acidic condition. Okay, so the option here for this question is only A, which says the non-ideal condition or the condition which is not related to microbe is only option A. Okay, beta. So microbes also found in acidic condition. That is why they are known as acidophiles. Right? Clear to everyone? If is it clear, please use the thumbs up reaction which is present, which is given to you beside the chat. Okay, great then. So today we are going to cover the total 6 topics. That is, we are going to discuss the uses of the microbes in household products, in industrial products, in sewage treatment plants, in biogas production, as biocontrol agents and biofertilizer. We are going to discuss each and every topic in a detail. Okay, so these are the topics for our today, and please get focused and cheer yourself up with a high energy so that we can have a good understanding. As you know, we have the microorganisms, which we call the microbes. They are basically categorized into 4 categories, right? We have categorized them into 4 categories initially: bacteria, fungi, protists, and virus. Okay, the bacteria, fungi, protists, and virus. See, here we have not taken protozoans because we are talking about the ideal microbes, right? Here we are talking about the human welfare, the microbes which are good for the human, which are helping them, not those are making the negative effect of them, right? So only we are taking 4 classes of microorganism here: bacteria, fungi, protists, and the virus. Okay, so as you can see here, what you can see, in first picture you can see a cylindrical shaped bacillus is there. See, the prions is an infectious RNA of the virus, as you have studied in your biological classification, right? So they are harmful. they causes the disease in the brain. They inflammate the cells of the brain and leads to some severe infection and the disease. Okay, so prions are not the microorganisms. They are basically infectious RNA. Clear? So then we have the bacillus, or we can say the rod-shaped bacteria. These rod-shaped bacteria are known as the bacillus, whereas the spherical-shaped bacteria are known as streptococcus. Examples we have the streptococcus cerevisiae, the streptococcus sacae. Several are there, and these rod-shaped bacteria sometimes they have the flagella, the hair-like, long hair-like segment, which helps in the movement. The flagella there is helping in the movement of the microorganism, right? So above three pictures are depicting your bacteria, whereas the lower three pictures, which is given as here, these are the viruses. The first one is our commonly known as the bacteriophage, the virus which infect the bacteria. And as you know, this is the protein capsule which is given here. This is the protein capsule of the bacteriophage, or we can say the virus which encloses the genetic material. Okay, so it encloses the genetic material, and here the protein capsule or the capsid which is given here is of adenovirus, right? And these adenovirus are responsible for the respiratory infection. All the kind of respiratory infection we are having, they are caused by the adenovirus. And the last we have the rod shaped like virus. They are the example of this is TMB, that is tobacco mosaic virus. Okay, okay, can you tell me what is the genetic material of this TMB? What is the genetic material of TMB? We have done this before, right? Very good, RNA, right? So they have the genetic material that is RNA. And if the virus is having the RNA as their genetic material, then we can say to which class this virus is belonging? The class of viruses? The virus which is having RNA as their genetic material, they are belonging to the, no, not phages. They are belonging to the retrovirus category. Okay, they are belonging to the group that is known as retrovirus. Okay. Yes, reverse transcription takes place here. Okay, so now we are going to start with the function or the uses of microbes in household products. So as you can see, this person is having so many delicious food in his dish. Then we have the bacteria, and on the right side we have, on the right side what we have? What is given on the right side in the picture? Right, it is curd. So we always have curd with our food. You know, we are habitual. Whenever we have food, we want the curd, either a salty form or in a sweet form. We make raitas of curd. We use curd so many in places. We make shrikhand, we make kebab, or we make lassi with this curd, right? So curd is used everywhere. Now this curd is made by the bacteria which is known as lactobacillus or the lactic acid bacillus, that is labs, which you have talked about, right? Which you have given the name, Nandish. Okay, and this lab is used to convert the milk into the curd. Okay, this lab is used to convert the milk into the curd. Have you seen when your mama sets the curd? Have you ever seen your mama working in the kitchen and preparing the milk for setting the curd? How she prepare it? She initially take the milk Coagulation. Okay, let's see. So she initially takes the milk and warm it up, right? She never boils the milk. She warm it up to 50 degrees Celsius, right? And let it cool it down a little so that the milk reaches to the temperature of 35 to 40 degrees Celsius. Okay. When the milk reaches to that temperature, then mama add a small amount of curd, which was already present in our house. So that small amount of curd, which we are adding in the fresh milk, it is known as the inoculum. It is also known as the inoculum or the starter, because that small amount of curd is rich with lactobacillus bacillus, right? So whenever we take a nutrient medium, whenever we take a small amount of nutrient medium, which is rich in the microbes, then we call that small amount of nutrient medium an inoculum. Okay, so mama is adding a small amount of curd, that is, she is adding the inoculum, and after adding the inoculum, she stirs the milk very gently so that the microorganism can spread throughout the fresh milk. Okay, and then she let that pot or utensil at a side of a kitchen undisturbed for 2 to 3 hours, right? What happens during that process? How that milk get converted into curd by the lab? How it is taking place? No fermentation is there. In fermentation, you see, the bubbles are formed, but there is no bubble in the curd. And that the milk contains a protein. Milk contains a protein, that is casein, right? The casein protein is digested. It is digested by the bacteria, right? And when it digests, the milk get set. You know, the protein get denatured, and while this process, they also convert the lactose sugar present in the milk into the lactic acid. That is why the curd is little flared. Okay, so this curd is not only used to increase the flavor of a food, but it also helps in the production of vitamin B12. The lactobacillus which is present there, it helps in the production of the vitamin B12. Initially, beta, this casein structure get denatured. Okay, it is not converted into anything. The structure got denatured due to the production of acid, right? So the casein become completely deformed. That is why the curd is forming. Okay, now this B12, the vitamin B12 helps in the formation of RBCs. These are responsible for the production of RBC. So whenever a person is having pernicious anemia, right? If the person is having pernicious anemia, that means the person is having the deficiency of vitamin B12. So if the person is having that, he is prescribed to eat a good amount of curd so that the vitamin B12 deficiency recovered and the person also recover from the pernicious anemia. Okay, so now we'll talk about now let's go and talk about the fermentation. See, the fermentation is another process which is also the breaking down of sugar, but in anaerobic condition, right? The setting of curd is taking place in a aerobic condition, right? But the fermentation always takes place in anaerobic condition, right? It is used to make the batter of dosa, idli. If you have ever had the bhatura, right? So the dough of the bhatura is also fluffed. The bread which you eat, they have so many pores in it, right? So what happens? During that, the bacteria converts the sugar into alcohol, converts the sugar into carbon dioxide and alcohol, right? And during this fermentation process, a lot of carbon dioxide is released. So whenever we heat the dough, the carbon dioxide start evaporating, right? It started evolving from the batter, but it get entrapped within. because the upper layer get completely hardened, so it get entrapped, and due to the entrapment of CO2, the product which is fermented become fluffy and has so many pores in it. As you can see here, see, this bread is having so many pores, right? So this fluffy texture is only because of the presence of the carbon dioxide which get trapped within it, and it is done by a fungi. It is done by a fungi that is known as yeast, right? So whatever yeast or the fungi we are using for the baking process, is known as baker's yeast, and the common baker's yeast is Saccharomyces cerevisiae. That is Saccharomyces cerevisiae. Okay, the same is present in the dosa batter also. That is Saccharomyces cerevisiae. That is yeast. Okay. Now, apart from that, we also use this yeast for the fermentation of liquid, like the formation of toddy, right? In the southern Indian portion or in southern states, you have find so many palm trees there. So you can see these black color pots hanging over the palm trees. So in that, they collect the palm juice, okay? Overnight, they collect the palm juice, and then they transfer that juice into the container in which the fermentation takes place. And when the palm juice get fermented, it get converted into toddy. It is a traditional drink in the southern states. Okay, you easily found the toddy everywhere. Same thing happens to use for the fermentation of fishes, fermentation of bamboo shoots and soya bean. Okay. No, now you can see. Have you ever had cheese? The Amul cheese, the pizza. You always like the double cheese pizza, or pizza has to be cheese burst, right? Jab tak usme se, whenever you not find something elastic like this coming from the pizza, we'll not enjoy it, right? So the cheese is also formed by the fermentation process, and for that we use a specific kind of the fungi for the specific kind of cheese. So this is the very common cheese we have seen, and this is the favorite for Jerry. Have you ever seen the Tom and Jerry cartoon? So you always find such kind of cheese present, and you can see the Jerry is also here. Okay, a relative of Jerry is also present here. So this is a Swiss cheese. This production of carbon dioxide gas, and it is produced by the bacterium Propionibacterium shermani. Okay, the Swiss cheese is produced by the bacteria that is Propionibacterium shermani, whereas the Roquefort cheese, or we can say the blue cheese, here it is the Roquefort cheese. It is made by growing a fungus that is Penicillium roqueforti, right? It gives a slight pungent flavor and smell to the cheese. It is very favorite cheese for the people in our outer countries. In India, it is not found usually. So this is also known as blue cheese. This you can see the blue and gray lines over the cheese. These are all the growth of the Penicillium roqueforti, which is giving it a pungent flavor. Okay. Now your question is, no, it is not spoiled. It is actually made to increase the flavor. You can find 90 different species of 90 different types of the cheese in there. Okay. So as you have already given your answer in the chat box. Okay. Guys, try to wait for 5 to 10 seconds so that I can launch a call. Okay. So, right, you have given a correct answer that the gas which is responsible for the puffed up appearance in the dough is carbon dioxide, which is produced during the process of fermentation. Right. Now we are moving towards the next topic, that is the microbes and industrial products. Now here we are going to produce the products at industrial level, right? So at industrial level, the fermentation of takes place. Fermentation of beverages takes place. So when we brew the alcohol, like we are brewing the beer, at that time we use the yeast, that is also Saccharomyces cerevisiae. Okay. When we are providing it a solid or a semi-solid medium, it is known as baker's yeast, because we are baking that stuff. But here we are brewing, here we are fermenting the liquid, so we are calling it as brewer's yeast. Okay. And brewer's yeast is used for the production of beverages by fermenting malted cereals and fruit juice to produce the ethanol, right? Whereas the wine and beer are produced without distillation. Okay, so here for each and every specific time of beverage, the alcoholic beverage, we use the different types of bacteria and the yeast. Next, the most important use of these microorganisms in the industrial level or for the human is the production of antibiotic. What is antibiotic? Can anyone tell me? What are antibiotics? What are antibiotics, dears? No, Nandish, there are two terms, beta. One is antibody. Okay, one is antibody which fight against the microorganism. It fight. It is a protein, okay, which is produced by the B cells or B lymphocytes, right? But here we are talking about the antibiotic. Antibiotic is a chemical substance, right? It is a chemical substance which inhibits the growth of the microorganism, right? It is not fighting against the microorganism. Instead, it is killing the microorganism. So antibiotics are the chemical substance which are produced by the microbes itself, but they are acting as the inhibiting agent. Inhibiting, that means killing or stopping agent for the related pathogens. Okay. So whenever we get infection, doctor always gave us some antibiotic. It can be amoxicillin, azithromycin, ciprofloxacin, right? If you have ever checked your prescription, so many antibiotics will be given. So depending on the type of the microorganism of infection, the doctor prescribe different types of antibiotics. Okay. And this antibiotic, the first antibiotic was discovered by the Alexander Fleming through a fungi that is known as Penicillium notatum. Penicillium notatum was a fungi from which the Alexander Fleming firstly discovered the antibiotic, and that antibiotic is penicillin. Okay. Right. Actually, what happened? He was culturing the plates for some bacteria. The Alexander Fleming was culturing the plates for bacteria. Culturing the plates means he is trying to grow the bacteria on the solid surface of the nutrient medium. Accidentally, the contamination takes place. Contamination, that means some another microorganism also started growing. So, okay, I can show you here. See, this plate, this is a plate, okay, on which we have poured our solid medium, okay, and in that we have put some bacteria so that they can grow and increase their colony size. These red dots are the bacteria. Okay. So what happened? He saw that the process he done, it was not done in a sterile condition, due to which a small patch of another microorganism appeared. So this yellow patch, which is I am making here, it is a patch of fungi. Okay, it is a part of a fungi. It is a colony of a fungi. So he observed that where this fungi was growing, none of the bacteria was growing in the close proximity. right? In the close proximity, none of the bacteria is growing. That is because of the production of an enzyme, right? That fungi was making a particular enzyme which is killing the nearby bacteria, due to which the bacteria were not growing in the near proximity of that fungi. So this is how Alexander Fleming has concluded that the fungi and that fungi was Penicillium notatum, that the fungi is extracting an enzyme which is killing the bacteria. That is how he called it. Yes, yes. The if you have heard the news in second wave of COVID, have you heard about the black fungus is there, yellow fungus infection is there, white fungus infection was there? Have you heard about it? So these fungi spores are present everywhere, right? It depends on they are getting a good condition, a favorable condition for their growth or not. So what happened during the COVID infection? For example, during the COVID infection, what happened? The patient's immune system get highly suppressed. Okay, the patient immune system get highly suppressed. So when he inhaled the spores, the spore reaches to the lungs of the patient, right? Or it reaches to the some other part of the body through the food, or you can say by the drink, right? But mostly the patient of COVID patients were having the lung fungal infection, because the mask which they are using for oxygen, right, they were not properly sterilized, right? So due to which what happened, due to which the spores went inside the human body, and human body, as you know, the lungs are very moist and the temperature remains 35 to 40 35 to 38 degrees Celsius, right? And that is the favorable condition for the fungi. So that is how the fungi start growing. The aspergillus is the fungi which usually causes the respiratory infection. Okay, is it clear, Nandish, now? Okay, so here are some bacteria through which we have used the antibiotic. Very good. Okay, so you have to write down, or you can take the screenshot if you have a phone. These are the names of the bacteria for which a particular antibiotic is used. Okay, now here we have the another table. Now this table is saying that not only for the production of the antibiotics, we also use the bacteria or different types of microorganisms for the production of the acid. As you know, the acids are found in two different forms. They can be organic or inorganic. If they are organic, they are present, or they are, can be produced through the animals or the plants from the living organism, right? But the non or inorganic acids are always produced in the laboratories or the factories. So here we are talking about the organic acids. Here we are talking about the organic acid. So the citric acid is produced by the Aspergillus niger, which is a fungus. Acetic acid is produced from the Acetobacter aceti, which is a bacteria, right? Butyric acid is produced from the Clostridium botulism, and the lactic is from the Lactobacillus. So these are some acids, and these microbes are the source of their production. Okay, so as you know, for the formation of alcohol, we have used yeast, right? That is Saccharomyces cerevisiae for the production. Apart from that, we also extract some enzymes from these microorganisms like lipase, as it having a suffix ase. Ase is always means, yes, brewers ase is always means to cut. Okay, the suffix ase is always used for a cutting enzyme, right? So here the term is lipase. So lip is used for the lipids or the fats. ase is for the cutting, right? It's for the cleaving. So the enzyme is lipase. That means these enzymes are used for the removal of the oil stains or for the decomposition or the degradation of the oils. So these lipases are used in the detergents, right? You can see whenever we have a oil stain on the cloth, mama always soak the cloth in a detergent. They never use the bathing soap over there because the detergents having that lipase enzyme which cleans or easily remove the oil stains, right? Then we have the pectinase and the protease, which are used for the cleaving of proteins. Okay, please write it down in your notebook that pectinase and protease are the enzymes used for the cleaving of proteins. Why we use the proteinase enzyme? See, in biotechnology, during the isolation of DNA, we have used the protease enzyme, right? And when we extract the juice, right? Initially, when you prepare the fresh juice at the home, that is not transparent. But whenever you find the juice in a packed Tetra packs, right? You always found the juices completely clear. You can see through it, right? So that is done by the removal of protein. Whenever they pack the juices in Tetra packs, they always add the protease enzyme so that the protein get denatured and that settle out, and all the supernatant was used for the packaging. In Maza, they are not using the protease enzyme. Okay, in Maza, they are not using it, but we can say it is using in a small amount in fruity. You can see the difference between the fruity and the Maza. Have you observed it? Maza is more translucent as compared, or we can say opaque as compared to the fruity. Okay. Next is we have the Repto Kinase. This Repto Kinase is also produced from the Streptococcus and it is used as a clot buster. It is, we can say, a kind of a lipase enzyme, right? It is used to remove the clots. You know, whenever you have the high cholesterol levels, the cholesterol get accumulated in the blood veins, right? In the blood veins and forms the clot. So those clots are removed by the enzyme streptokinase. Then we have the cyclosporin A, which is produced from a fungus, that is Trichoderma polysporum, and it is an immunosuppressant agent. See, whenever we are doing some transplant procedure, why we need to, okay, let me tell, can you please tell me why we are using the immunosuppressant agents during the transplant? Yeah, steroids it is. Why we are using immunosuppressant during organ transplant? Right, to suppress graft rejection. And how that graft rejection is taking place? See, we are transplanting a human kidney to another human. Why it is getting rejected? Right, by our immune system, because that kidney, if we go for the kidney transplant, which we have taken from the donor, is not exactly the part of our body, right? So our body is very clever, very smart. It can easily recognize that, yes, this organ is not having the tissues which are the part of our body. So they start acting on that kidney and destroy it. So to reduce that effect or to inhibit the destruction of the donor kidney, we use immunosuppressant for some time. Then we have the statins. These are also steroid. They are extracted from the yeast, that is Monascus and purpurus. Monascus purpurus. It is also used for lowering the cholesterol agents, right? Sorry. It inhibits the enzyme which is responsible for the synthesis of cholesterol. The person who is having the heart problem, who is having the high blood pressure issues, they are always given some statins. If you look if you're in your family, someone having the high blood pressure, they having the atorvastatin, right? The statin is there to thinning the blood, and that thinning of blood is only takes place by lowering the fat levels. Okay, so here is your question. Let's see. You can give the answer of this question or not. What is the difference between blood cholesterol agent and see, the cholesterol level is all about the presence of fat in your blood. But when we are talking about the clot formation, when the clot formation is taking place along with the fat, along with the cholesterol, the platelets, the red blood cells also get accumulated, right? So that lipid clot started having a coagulation along with the protein. So that clot is not only having the cholesterol, that is also having a small amount of platelets and the fibrinogens with them. Okay, so here is your question. The common name of Saccharomyces apilopsoidens is, yes, Yadagiri, you have given a right answer. The option is B, the wine yeast. See, the wine is not prepared by distillation. Okay, for it we are using the Saccharomyces ellipsidon. Brewer's yeast is Saccharomyces cerevisiae. Sake yeast, the Saccharomyces sake, and the alley yeast is Saccharomyces piriform. Okay, so these are the different types of we can say, sorry, the fungi, right? So next is the treatment, the sewage treatment, which is a great issue nowadays, because due to the increasing population, the sewage amount is also increased, right? So the sewage is considered as the municipal waste. We have talked about it in our the last chapter of ecology, that is environmental issue. The sewage is a municipal waste which is coming from the houses, which is coming from the industries, and it basically contains the organic matter and the microbes, like the fecal element mostly, right? And this water, the sewage which is coming from the houses, right, they have to be treated initially because they contain the pollutants as well as some harmful microorganisms. If we release this sewage water in our rivers and lakes, they will contaminate the fresh water, right? So we have to go for their treatment. We have to go for their clarification so that it will not harm the fresh water resources. So that is STPs, that is sewage treatment plants are made, and this treatment takes place in two stages, in two different stages. First is primary treatment and second is secondary treatment. The primary treatment is also known as the physical treatment. It is also known as the physical treatment. Let us discuss why this primary treatment is called as the physical treatment. So why we are calling physical? Because we are using the physical methods to remove the particles, such as big particles. We are removing here the pebbles, the rocks, the wrappers which are present in it, the plastic bags which are present in the water, right? You can see everywhere the person usually drops their garbage in the water. So all those floating mud debris are separated by the filtration. So that here we have the sequential filtration. We use so many sieves. Sieve, you know about sieve? A plate which is having so many holes. Mama, whenever make the rotis or chapati, they always sieve the flour before making a dough, right? So we have so many sieves here. You can see the 5 to 6 sieves are here, and each and every sieve have the different sizes of hole. The initial one is having the large hole so that the larger particle get separated. As we move towards from left to right, the sieve size keep on decreasing according to the particle size. So this is how we are separating or we are filtering the floating debris. Okay, after that we are left with the water which is containing the soil and sand mixed with that water. So soil and sand can only be separated by the sedimentation. You have studied about the sedimentation. When we leave the water undisturbed, all the sedimented particles, all the soil particles, sand particles started sedimenting under the force of gravity. Okay, sorry, so we are removing the grit, that is soil and small pebbles, by the help of sedimentation. And when the sedimentation takes place, you can see the sediment is present here. The rest, the supernatant, is transferred into the next tank. Okay, they are transferred into the next tank. Now here, the settled solid which is formed here is known as primary sludge. This is your primary sludge. Okay, and the supernatant which is formed here, which we are transferring from one tank to another, it is primary effluent. Okay, so we have two different names. Effluent is for the supernatant liquid and the sludge is for the sedimented particles. Okay, please keep this thing in mind. We have done the primary treatment, so whatever gets sedimented, it is primary sludge, and whatever is present in the liquid form, in the supernatant, it is known as primary effluent. Okay, now we move to the secondary treatment. In secondary treatment, what we do, we have a tank which is having the rotator. Okay, it is having the fan or turbine type rotator present in the middle of the tank. It helps us to circulate the air throughout the tank. This process is also known as aeration. Process is also known as aeration. That is, we are keep on maintaining the amount of air throughout, or we can say the oxygen throughout the tank. Why we need to do the aeration? Can anyone tell me why we used to use the aeration for the primary effluent? To help. No, it help in aerobic respiration, beta. And these, we are providing air. So whenever the air is present, the process is taking place aerobic. We have the aerobic respiration there. Okay. So in this tank, in this aeration tank, along with the primary effluent, we add a small inoculum. Yeah, right. We are adding some aerobic microbes in the form of flocks. Flocks, that means, okay, it's no problem. The flock means the small sizes. Okay. So here we have the bacteria associated with the fungal filaments. They are forming a mesh-like network. They are provided to this aeration tank, and when they are added into the aeration tank, they start degrading and decomposing, or we can say the breakdown of the organic components. Okay, they start breaking down of the organic matter into the inorganic. You have studied the decomposition, right? So when these bacteria acting on the organic matter is now the food for this bacteria and the fungi. So when they keep on growing, the amount of oxygen also increases, because they need a good amount of oxygen for the increased population. By log zyada honge, when we have the good amount of population, we require more oxygen to breathe, right? In the same way, as these aerobic bacteria increases their number with the increasing organic matter, the oxygen demand also increases. So that increased oxygen demand is known as the biological oxygen demand. This biological oxygen demand shows us that how much organic matter, that means how many contaminants are present in that primary effluent, or we can say the sewage, right? So the biological oxygen demand, it is the amount of oxygen consumed by bacteria during the breaking down of organic matters into the inorganic ones. And here we are talking about the 1 liter. We always measure the BOD in a 1 liter of water. So as the BOD increases, the high amount of BOD always says the high amount of organic matter. But when the BOD keep on decreasing, when the oxygen demand keep on increasing, we can say, yes, the water is getting purified. The organic matters are also decreasing from that polluted water, or we can say the sewage water. So here it is written, greater the BOD, the more is its polluting potential. Okay. Okay. After this whole, we can say the degradation, decomposition of the organic matter, we transfer the secondary effluent into the final settling tank, a sedimentation tank, because now these bacteria and fungi started dying. So they started getting settled because they no more have food, right? They no more have food to eat. So they started settling down. And whatever is left on the upper side, whatever is left on the we can say the supernatant, it is released into the nearby water resources, either it is lake, canal, or a river. Okay. And whatever gets settled, right, the microbes which get settled are known as activated sludge. Now this activated sludge will be used as in a small amount as the inoculum for the next batch. Now we use this activated sludge in a small amount again for the next batch, so that the next batch of water get completely clean. Okay. Is it clear to everyone? Yadagiri, Nandish, is it clear to you, beta? Okay. So here we have some anaerobic bacteria as well. If you know, we have produced the sludge, right? We have produced the sludge. We also uses the anaerobic bacteria for the sludge digestion, which is usually used in a biogas plants, right? If we have the organic matter in a solidified or the semi-solid form, like in a slurry, right? In the rural regions, the villages, people, or the we can say the farmers usually have the cattle in their farm, right? So those cattle produces the dung, the cow dung is there, the human excrete is there. They make a slurry of that and put it under the underground tank. Okay. They put that slurry in an underground tank and cover it, completely seal pack that tank. While doing that, you know, the cattle dung, or we can say the cow dung, contains the methanogen. Methanogen is an anaerobic bacteria which is present in the gut of the cattle, right? So yes, gobar gas is produced there. So what happened? These anaerobic bacteria keep on growing, and anaerobically they digest the organic matter into the inorganic. Okay, so now they are converting the slurry, the sludge, into the manure. And while this conversion, they produces a good amount of gas, and that gas is the collection of methane, sulfur hydride, and the carbon dioxide. And the collection of these three gases, combination of these three gases, is known as biogas, which is used as a fuel supply for the household work. Instead of using LPG, these people uses this methane, sulfur hydride, and carbon dioxide, that is biogas, for their household purposes, right? And the sludge which is formed, a solid which is formed at the last, it used as a manure. It is a very good and nutritious stuff for the crop. So instead of using fertilizers or the chemicals, we can use that organic matter for our plants to grow and get a good amount of nutrients. So this is the overall a procedure of this, we can say the biological treatment, right? Or we can say the secondary treatment. Okay, if you want, you can take the screenshot of this. Okay, so now moving further, this is the whole sewage treatment plant. These are the different steps. As you can see, these plants are placed in acres of land because they require a good amount of space, because the sewage which is produced is also in a very great amount. Okay, so due to the non filtration sometimes of the sewage, the Ministry of Environment and Forest has initiated the plans of Ganga Action Plan and Yamuna Action Plans, because the industrialization has increased the release of this polluted water without any treatment inside the river, which is contaminating the river day by day. Okay, so here is your question for today, the last question for today. Which of the following is not correct regarding the primary treatment of the wastewater? Your option says A is initially floating debris is removed by distillation. Option B, the grit and pebbles are removed by sedimentation. Option C, solid that settled from the primary sludge. And the D says the supernatant forms the effluent. Okay, so give your answer through this poll. I'm launching the poll here now. You have to give this answer in a poll. Yadagiri and Nandish, please give your answer in the poll. Okay, very good, everyone. So you have given your quick response. So as you can see, everyone has opted for the option A. Very good. So option A says the initial floating debris is removed by distillation. No, we are not doing distillation. Instead of distillation, what we were doing there? Which process we have used? Sieving, and that sieving effect is used in the process of filtration, right? The process is filtration. Okay, dear, the process is filtration. Sorry, for the T, it is L, filtration. The process is filtration, in which we are using sieves, or we are using the sieving effect. Okay, so this is all for today. From tomorrow, we'll discuss the microbes in the production of biogas, and we also use their discuss about their biocontrol agent and another stuff. Okay, so before leaving the class, please give your response to this NPS poll, and till tomorrow you have to read your NCERT, go through your NCERT very well, and do practice all the module questions related to these topics which we have covered today. Okay, so before leaving the class, please give your response or feedback in this NPS poll, and after that you can leave the class. Okay. Okay, thank you so much for your precious time and the feedback. We'll meet tomorrow with the continuation of topic, and okay, and we'll definitely complete this topic tomorrow. Okay, till then, bye and good night.